So we have a question about the cytoskeleton. Could you explain the difference between centriole and centrosome? Centriole is the part of the centrosome. So it's just a structure that forms the centrosome, because centrosome is the generalization of all these structures. And when we didn't have very advanced techniques to kind of take a closer look at the structure of the centrosome, scientists from all parts of the world, they started investigating it and started naming it, naming it as they wanted. So some people started naming it centriole, others centrosome, and in the end, when they defined smaller parts of the centrosome, they decided to give the name of centriole to these long 9-unit structures. So these 9-unit structures are centrioles, and they, both of them, and the filaments, they form the centrosome. Okay, so we have two questions, and the first question about the cytoskeleton of the organelles. So the only organelle that has the cytoskeletal structure is, it's not only, but the biggest one is nucleus, because it is inside the envelope, and this envelope is powered or forced with the cytoskeletal filaments called lamins, and they make it rigid, so nothing can bend it, nothing can tear it. That's why these filaments form kind of protection for the nucleus. And as I said, some of the organelles are fixed in space, and it means that these, just a second. Let's say you have a cell, and you have mitochondria or Golgi apparatus. So if you don't have something to keep them in the place, they will be floating inside the cell. So one mitochondria to this side, another to here, and everything would be floating freely. And we actually need to make some compartments of the cell that will be doing some function according to the place that they are located at. Let's say you have a neuron with a huge axon, and this neuron needs to really concentrate some of the vesicles in the axon, in the terminus of the axon, so some neurotransmitters. And some of the structures need to, organelles or anything, they should be in dendrites, for example. So to be able to separate these compartments from each other, we need some kind of mesh that will hold everything in its place. So let's say this small mitochondrion, it is kind of glued to these two filaments, and it doesn't move because of these filaments. And whenever the cell needs it to be in the other place, it takes it, and with the help of motor proteins, it starts moving it across but the movement is very controlled. Without the need, the cell will not take this mitochondria from this place, and it will not change its location. That's how you may concentrate, for example, mitochondria in one place that really needs energy for production of the proteins or movement, etc. The same happens in sorry. The same happens in neurons. So because of this mesh, we can concentrate the neurotransmitters in the axon and some mitochondria to receive and send the signals in dendrites, for example. And this is why we need the cytoskeleton mesh that fixes the organelles in their places. Okay, so intercellular transport. So you have to understand one thing. So let's say we have 3 cells and intercellular. So inter means from one cell to another, and it's just between 2 cells. And if you think about insides of the cell, so from one part to another, it will be called intracellular. So which one you asked? Was it intercellular or intracellular? Inter, okay. So intercellular is usually having, okay, so it's 3 types of the interaction. So one might be paracrine, autocrine, just a second, endocrine. So these 3, I'm not really sure if they're called like this, but I remember them like this. So all of them are related to the intercellular interactions and intercellular transport. So usually we mostly have paracrine and autocrine processes that happen to transfer some vesicles between the cells. So autocrine means the cell secretes something and then consumes it. Usually it's needed to kind of tune itself, just to kind of control the processes in a manner that you secrete few molecules that will be obtained by the receptors. And these receptors, so they take these molecules and they start a huge signaling pathway that changes everything inside the cell. So this will be one part of intercellular, but also other cells in the in the close proximity, they can also take these substances and start the same signaling. And so this is paracrine and autocrine. And if you think about the transportation itself, so intercellular. So let's say one cell produces the nutrients that the other cell does not produce. So we have nutrients plus and nutrients minus, for example. So this cell produces these lipids, for example, in the smooth endoplasmic reticulum, and forms these vesicles with this lipid, and it starts moving it to the biomembrane. And after that, it secretes them, and the other cell catches these vesicles, so it starts the endocytosis of these vesicles. That is the intercellular transport. Question about lecture 4. Can you please explain the difference of bright field and dark field? Yes, just a second. But I guess this question was on one of the previous Zoom sessions. Okay, so in the bright field and dark field, we have the same set of objectives, but we have different illumination of our samples. So let's say this is a cell. And in the case of bright field, we have parallel rays of the light that come to our sample, and they usually come under the right angle, so it is 90 degrees. In this case, the light usually passes through whenever it doesn't have any obstacles. So in this case, for example, very close one, because of the thickness of the cytoplasm, it will start bending a little. So this part of the cytoplasm, it will look much darker, or a little darker than the other thinner part of the cytoplasm. Whenever we have, whenever we have rays coming to a solid nucleus, for example, this light is scattered, and we lose this part of the light beam. So this is the light beam. And for example, this guy will also be scattered. So this one will bend a little, and this guy will still go in a parallel way. So in this case, the objective will take or will catch only the parallel rays, and we will see something like, so this is what we see. So something like this, and this part will be very dark, and since this part is bending a little, so this will be darker than this part. So this is super bright, this is bright, this is a little bright, and this is dark. So this guy is dark. And this is the bright field microscopy. Everything goes in a parallel way, and we lose the scattered light. In the case of dark field microscopy, we intentionally start illuminating under the angle. So we have this parallel light beam coming like this. So this is the light beam. and these light rays, they come, let's say, to our cytoplasm. So and we lose this part of light beam. We don't catch it with our objective. But whenever we have thicker parts or very solid ones, some of them will be scattered. So this guy will be scattered in this direction, but this guy will be scattered towards our objective. And for example, this one will start bending and also will be scattered towards our objective. Other parts will just pass through the cell and we will lose it. That's why when we look at this part, what we see is actually everything dark. And the ones that were scattered towards us, they will be little bright, and the solid ones will look very bright. So it will be super bright. And here everything else will be very, very dark, because we lost all of this background. That's why it's called dark field. So most of the image will be dark, and here most of the image will be bright. And this thing, like this window, is called field. And in this case, it's field of the, again, dark field and bright field. Another question. How do actin filaments work in the cell? Camilla, so they are polymerized and they form some kind of mesh. So mesh is this network of filaments, and this mesh is kind of inside the cell. And and if we have motor proteins, they can start moving across. So let's call these guys actin filaments, and this is the motor protein. So it kind of binds to the actin filament and starts moving across it. And if you have like many filaments that are interlinked, it can move from one filament to another. So it's a kind of path for this motor protein. And whenever it moves across it, it kind of gets to the place that it needs to be at. And since the whole cell is kind of holding this mesh, it means that you can get from one part to another easily. But to move these motor proteins, they use ATP. And this ATP is, as I said in previous lectures, they hold the energy and they can easily share this energy. So it's a energy dependent process to move from one part to another with the motor protein.